talking at different time points as we go along. So the topic of the talk, as I already said, microbiome research in the age of Corona. I'm sorry that everything these days is about Corona, but we just need to have one meeting, I think, at least focusing on this topic also for this topic, which is a bit different than what's going on in the clinic. So, and I'll just talk you through the plan before Marius takes over. Brief intro about the COVID-19. We have variable background on this, so just a brief intro would be good. And then the big question really is gut microbiome affected by COVID-19? And some questions related to this. Why would we think so? Is there any data out? What kind of research questions do we have? And how does it affect us really? And what else can we do than what we usually do? And then discussions as we go along and in the end if we need to. Okay, Marius?
Yeah, thank you. I'm a microbiota researcher, but my main position as a consultant at the infectious disease department at Rikshospitalet. So there we receive some of the worst cases of COVID. So I'll just give a very brief introduction. So this first slide shows the virus. It's called SARS-CoV-2. So it stands for Severe Acute Respiratory Syndrome Coronavirus. So we have known about coronavirus for years. It's a virus that can cause disease in animals, in humans, and in birds. It's structurally not very small. It's an RNA virus, and it has a lot of these spike proteins on the surface, which give it a crown-like shape. So that's why the name. So we have had outbreaks of coronavirus before. We had the SARS, so the SARS-CoV-1 in the early 2000s, and then we had the MERS, the Middle East Respiratory Syndrome, a decade later. So back in those days, we were really worried about the pandemic, but it never happened. So for some reason, this SARS-CoV-2 has got pandemic potential. And if you have a look at the lower part of this figure, you see that one of these spike proteins is attaching to human ACE2. That's the angiotensin converting enzyme. We know it from drugs that treat hypertension and heart failure. And for some reason, these viruses are using this as the receptor to enter the cell. So next slide. So after a variable incubation period, some patients will be asymptomatic, others will get symptoms. And the classical symptoms, as you read about, is fever and respiratory symptoms. But it's very variable. I tried to set up a case definition for this when I worked in Africa this winter, because it's so difficult to start triage on this. But I mean, there are so many patients showing up with fever, so many with cough, and there are so many COVID patients without fever and cough. So it's really a bit difficult to tell. If you go back a bit, Johannes. But there are also other symptoms like fatigue, anorexia. We have these strange symptoms with changes in smell and taste. And very recently, there have been some clusters of children with some hyperinflammatory syndromes, which affects the gut and the heart and the skin. So this it's a bit like Kawasaki syndrome. So it's a strange virus. And also after a few days, most patients get better, but then some really get worse, and we do not really know what that happens. And then they come to us. So next slide. Yeah, so you have followed, of course, the course here, and we have seen that the epicenter of the pandemic has shifted from China to the Middle East area, South Europe, and now it's really spreading out in the US and especially in Latin America. Next slide. And as you know, in Europe, it's really a few countries which have been really hit hard. It's France, Spain, Italy, and UK. And we also see large differences between Norway and Sweden. Next slide. So this virus, it uses this spike protein to attach to ACE2. to get in touch with the cell, and then it's cleaved by a protease, and it probably enters the cell with the endocytosis. The RNA virus is liberated, and then it started to reproduce. It makes structural proteins and new viruses. And this figure is interesting because we can attack pharmaceutically on these points.
Yeah.
Next slide.
Well, yes, and I guess what is this is probably a very important to know the details here, because the big question is really, why is some sick, why are some not? And one of the things is really going to viral load. You know, how much exposure do you have? It could also be matters related actually to how much replication do you have and things like this. So, but we will know more hopefully, because there's so intense research activity going on. But so that was a general intro, and then the big question for us here when you talk about microbiome is the gut microbiome affected by this disease? And I guess the first question then is, why would we think so? And I think that's an important question. And the main reason is really that there are symptoms from the gastrointestinal tract. And I think the way I think about microbiota is really that when there is something, when there are symptoms from the GI tract, then there is probably at least changes in the microbiome. Whether that's really relevant for disease or severity, we don't know, but it's definitely could be relevant. And this is from not a big study, 204 patients from American Journal of Gastroenterology, where in this cohort at least, you had quite a lot, 47% with both digestive and respiratory symptoms, and even some patients with only symptoms from the GI tract. And this is from a review, systematic review and meta-analysis actually, and that was also just out in Lancet Gastroenterology Hepatology. I think it's very interesting to see actually that this is the field where there are so many papers out. Most of them are not reviewed yet, and people are already writing the meta-analysis and the systematic reviews before we have even seen almost the original research. But at least here we could summarize 6,000 patients, and you have more moderate numbers, but it seems like at least 15% have digestive symptoms, but wide ranges in the studies from 2 to 57%. And you have diarrhea, loss of appetite, etc. And abdominal pain. It's known that the acute abdomen, that's just stomach pain, could by itself be caused by COVID-19. So there's definitely something going on in the gut. And the other thing is that it seems from studies that, from this review also, that those patients that have severe or critical COVID, actually they have a higher prevalence of gastrointestinal symptoms. So whether this is because something going on in the gut is also driving severe disease, or whether these just operate together, we don't know. But it's interesting observation. So I think one important question also is why does the virus go where it goes? So what's the tissue? What determines the tissue tropism? That's why do the virus infect the lungs or the gut or whatever? And looking at the main entry point here, which is then this ACE2 from the Human Protein Atlas, it was possible to produce these figures, and you can see actually how the gut is really screaming against you, right? So it's very strong protein expression in the small intestine. And I'm not. If you look a bit more at the figure, you realize something is missing, and that's the lungs. So this is the most severe cases really have bad lung affection. But why is there no ACE2 in the lungs? And I think this also says something about the weakness of the Human Protein Atlas, because it's definitely there. It's been shown many times that ACE2 is expressed, but it's not necessarily in all the cells, or it could be methodological issues with antibodies and so on. This is just from a paper in Science where they use organoids, where they grow. This is lung epithelium, which is grown as in vitro organs, and they infect them with SARS-CoV-2. They show, I don't show everything here, but they show that ACE2 is expressed in the lungs, and these reds here are actually infected then with SARS-CoV-2 virus. And here they compare it with the original SARS-CoV. that was in 2003. So both infect pneumocytes in the lung. So, but from the same paper, which was the main point of this paper, is actually that here they show that ACE2 is expressed in the small intestine, and then they show they infected these cells with SARS-CoV-2, and you see after some hours you get viral replication. These are the gray areas in these intestinal organoids, and they show them here with electron microscopy. So there's definitely viral infection in the small intestine and viral replication. Okay, so something going on in the gut, but does this have anything to do with microbiome? We don't know yet. But one interesting point that was clear quite early was that we will find the virus in the stool. And as you can see, this has already been reviewed, at least in one paper, where they have something wrong here, but they included 540 patients that were COVID-19 positive. So it was in the nasopharynx. And out of these, 291 had a positive fecal PCR for SARS-CoV-2. So more than half in this, just when piling up the studies and patients included in those studies. But very variable methodology. What time point was sampled from day 0 to day 17 of the first positive test. Another question then is, how does this last? Do you keep shedding virus? And based on the 199 reported patients who were positive in stool and were also followed up, more than 60% were still positive after some time, even after a negative nasopharyngeal swab. So, but the duration of the observation was very variable, so up to a month and so on. So we don't know really. We just know that it's probably there in about half, and it could last for a long time. But we have no idea whether it really causes disease, I think, or whether there are live viruses at all, or just virus remains and so on. So this is an important question that's important for us in the microbiome research world. Another thing, we're talking about stool, but this is actually from a paper that looked at whether saliva could be a relevant material for diagnosis of COVID-19. And you can see that it actually works quite well, that you have a high degree of positivity of SARS-CoV-2 PCRs in saliva compared with nasopharyngeal swab. So this is a potential in this. But I think nasopharyngeal swab and saliva are actually two fundamentally different things, or at least to some degree. Saliva, I think, is in principle a part of the GI tract. So it seems like this virus could reproduce actually in the entire gastrointestinal tract, more or less. So the gut is important. So the second question is, do we have any data on microbiome in SARS-CoV-2? And because there have been, if you read the papers and follow news, etc., there have been some headlines also on the microbiome in this situation. And one of the preprints, so it's not peer-reviewed, stories that have been cited a lot actually in the newspapers is this one: gut microbiota may underlie the predisposition of healthy individuals to COVID-19. But let me tell you, I'll try to convince you in one slide soon that this is not really data. We don't know anything about this. What we do see in this paper is summarized in this figure. They actually study blood in this study. They have 31 patients, 13 severe and 18 non-severe with COVID-19, and then they analyze proteins. They make a proteomic profile. And then and they find that this proteomic profile is actually then different in severe compared with non-severe. Okay, so they have a potential biomarker of severe disease. And then they go back to a study they did several years ago, before the occurrence of this virus at all, where they had more than 1,700 individuals where they have microbiota profiles and blood, so they could measure the same proteins in blood. And there they found that this proteomic profile associate somehow with some composition of the microbiota. So in healthy individuals, there's some association between some proteins and what's going on in the gut. There's really no data on the microbiota of the COVID-19 patients. And these proteins, they strongly correlate with inflammatory factors like CRP and IL-6. So I think it's really to push it to say that this suggests that the microbiota predicts severity of disease, because there are so many other things in this situation that really determines the degree of inflammatory markers. I found, I really tried to look around at preprint service and so on. I found one more story on something related to microbiota. This is from pharyngeal samples. And here they actually did, here they had the 46 positive COVID-19 and 89 negative ones. And then here they actually did some 16S sequencing, not a full unbiased panel, but a set of specific PCRs for a specific bacteria. And then they try to summarize this. The green ones are negative and the red ones are the COVID-19 positive. What you can see here is actually the different bacteria that have investigated down here, and each individual has one column. So you can see here, they try actually make some kind of cluster, and they claim this is a cluster, which is more similar. And you can see there are a lot of proteobacteria, for instance, in this cluster. And when you see here, then it seems like there are more green ones than red ones. So there are more negative COVID-19 negative that are in this cluster than positive. So this suggests that this is like a signature associated with COVID-19. But honestly, this is not very strong. So to summarize, I would say we still don't have any relevant data on the microbiome in COVID-19, at least from what I have been able to find. But please let me know if there is something I have missed. So perhaps it's your turn now, Marius.
Yeah, I mean, we have rehearsed this for hours, haven't we? Yeah, so far there are very few data on COVID and microbiome, but still there might be some research questions that are worth asking. So of course it's to get an understanding what is happening. Could the microbiota composition explain some susceptibility to infection or GI symptoms or the severe disease? Example, during the first wave of SARS infection in the early 2000s, patients who had fecal shedding of the virus, they had a more serious clinical course. So could this be partly due to the gut microbiota composition? And of course, if the microbiota is altered, is it possible to target the microbiota or microbial products to treat COVID? Is it possible to use it as a biomarker tool? For example, in these kids with these hyperinflammatory symptoms that also alter the gut? And is it possible to predict the treatment response? Next slide. Yeah, so as Johannes said, the intestinal epithelial cells, they express ACE2, the SARS-CoV-2 infection, and it's viral shedding and productive infection in the gut. And one research question could really be if this infection in the gut or the microbiota alterations in the gut could alter also distant organs like the lungs, the heart, and the brain. We know that a lot of people, we know that most patients get lung disease, but there are also a lot of patients getting symptoms from the heart and also alterations in the consciousness. Next slide. So in my previous career, I've been interested together with colleagues at Rikshospitalet and Ullevål in the gut-heart axis. And we know that in patients with COVID, maybe 20 or 25% of hospitalized patients have some kind of cardiac involvement. At least we see that they have some have alterations in the ECG, some have increased cardiac enzymes in the blood, and some get myocarditis, and some get really overt myocardial failure. So this is just a small study. There is an observational cohort that has been gathered from different Norwegian hospitals, and at the research institute at Rikshospitalet, we have analyzed some soluble markers that looks at different patterns of inflammation and so on. But I've asked if we could look at LPS binding protein, which could be a marker of gut damage, and also inflammasome activation, which is triggered by LPS. And to the right here, you see patients with cardiac impairment in red, and patients with normal cardiac function in blue. And we have measured these markers at 3 different time points, and you see they here at baseline. Patients with cardiac involvement, they have 3-fold higher levels of LPS binding proteins. And this difference is maybe not that high further out in the disease, but still it's quite striking. And we also find very close correlation between this gut leakage marker and markers of inflammasome activation. And these markers are also closely related to NT-proBNP and troponin levels. But this is, of course, just suggestive. We do not know what is happening here. Could it be that this ACE2, if you remember this picture by Johannes, ACE2 is expressed in the gut, but also in the heart, if you look at this proteomic atlas. So we are also running now an intervention trial. You have probably read about the WHO study. So here we are also biobanking. And we aim here to do echocardiography of all patients at 3 months follow-up, and then also to sample fecal samples to do microbiome analysis. Next slide.
There's a question from Brian here, Marius. Is there evidence that common or seasonal coronaviruses trigger cardiac episodes?
Once more.
If there is evidence that common or seasonal coronaviruses trigger cardiac episodes?
I'm not sure. I mean, seasonal coronavirus is mostly causing quite mild disease, so they are normally not hospitalized. And in the first 2 waves of SARS and MERS, I'm not sure. So it's not, you know, when we have patients with myocarditis or pericarditis, coronavirus is not one of the typical viruses that we analyze in the lab, so it's not very known.
Okay, one more thing here on microbiota research in this topic. Maybe this slide could be somewhere else, but we can take it now anyway. I think one of the questions that occurred actually, we can come back to that actually. Because after this, we were planning to think about how this virus actually affect our microbiome research. And this is really how it affected us, I would say. I'm not sure how this has been for everyone, but at Rikshospitalet, I guess you can appreciate the emptiness of the hallways. Our institute was completely shut down for 4 weeks as the country also closed down. We're luckily now opening, so it's, at least for lab work, it's more or less back to normal when it comes to activity, or at least almost, but as long as you, of course, you have to follow the guidelines. But I think one question is that that occurred to us quite early is whether the material is infectious. Can we handle it in the lab and so on? And I'm not sure we really have talked very much about that question. I
can just briefly say here, Johannes, that we were discussing in this treatment trial how to collect the fecal samples, and of course the gold standard would be to collect it like we used to do, but in this observational study there were already plans to do rectal swabs rather than the classic way of collecting. And the good thing there is that the patient can do this himself, and it's less exposure. But of course, there's also less material.
Okay, so you have the emptiness first, and then you have other things that came a bit of a surprise to me also. Of course, very early on, the world said there is really a shortage of reagents for diagnostic COVID PCRs. and there were, and there was, and there maybe it's still. And then the companies, of course, they changed their production lines. But when you do something, then there's something else you cannot do. And we just now realized that we have a major project where we don't have enough kits, and then it turns out they have stopped the production line for our favorite PSP spin stools kit. So we really do not know now how exactly yet how to handle a couple of thousand of stool samples waiting for us to work on. So, and also QIAGEN have changed their production line. So this is something to think about when you plan projects now, that you talk with your distributor and find out what's actually possible to get hold of. They say maybe we'll start up again in September, but who knows how the world is in September. I think also there has been some trouble with sequencing capacity, but this depends on the roles of the sequencer you use, whether they could have actually prioritized using virus-related activities and virus sequencing and so on, which has been of interest some places. So this is, of course, difficult. But then I think this one is another one that's very important in our field that changed many things. Because if the virus is shedding in stool, then what about fecal microbiota transplantation? And the FDA in the US, they published a warning in late March where they say you should not use those stools donated after December 1, 2019. That's mean after the occurrence of virus, because we don't know what's going on. So everything collected after that has to go into quarantine. And then what to do? It's actually been quite a lot of written material on this, so you can just search Google a bit and you'll find out what's been saying. For instance, proposals to do actual screening, and that's probably the way ahead. That's how we do it for all other infectious agents in this donation practice. And what is good is that the donor eligibility criteria is actually quite strict. So based on what's already guidelines, you would probably exclude patients that have symptoms that could be COVID-19. The problem is then, of course, those who do not have symptoms. And I think it's still not, we don't have the exact numbers there, how many are asymptomatic. So in this group from Birmingham suggests that from now on, we can only use the FMT products when we have a validated stool test for SARS-CoV-2. And this we will have very soon, I guess. There are lots of protocols out there, and this is being tested and so on. So this we will probably be able to handle, but definitely changing practice. And for us, we are trying to set up a donor biobank here, and all of this kind of has implications for what we can do, and it stops really initiatives that aim to build up new activities. And I guess this is the feeling of many people. It's not just research, of course. Remember, all the clinical activities have been locked down, surgeries, so many people suffering. But research is also suffering. Okay, we're back in the lab, but what about all the clinical trials? Many are put on hold. And how can we do this now on and just ahead? And FMT trials, not the least. So I think it's important. That's why I really like to mention it. It's important that okay, we can work and should work on COVID-19 because it's important. So we have to. It's really important to contribute where we can for such a critical situation that we are in the world, but. At least when we get on top of things, it's important also to actually keep pace of what we're actually doing, our original clinical and research agenda. So we not just switch everything, although there is an option to do something. In particular, if you have a very good research concept that works on many conditions, of course, tempting to test it on COVID-19. It's also possible to really have a brilliant idea. And then, but then I think you have to be early to get it out there, because I must say, you probably follow the news as well, and research news, the activity of the entire scientific community in this field has been amazing. You've seen all the molecular structures out there and everything we already know just a few months after the virus was actually discovered. But still, it may be that our research has stopped to some degree, and what can we do? And there are some examples out there. What can microbiologists do? Just some examples. I Googled some of those famous ones. Eran Segal from Israel is really one of the opinion leaders, and I write these days. He is the man behind, for instance, the big study on diet and postprandial blood sugar and the effects of bananas. And the banana is not the banana, whether, if you remember those talks. What he did, he really changed his computer guys to working on how to tools to detect outbreaks. So in Israel, they have really implemented a daily reporting system for everyone where you can report in symptoms, and they'll use this to detect local outbreaks. So this is very well published now, and an interesting way of using your capacity, I think. And then I searched for Rob Knight, definitely one of the kings in our field, and of course calling for citizen scientists to contribute, because he is a man supporting crowdsourced research projects, and they have this MicroSeta initiative, which is they have been running for some time, where people can provide a stool sample in a particular set, and then I think they have to pay $100 or something, or less or more, I don't remember. And then they get, of course, back their fully sequenced metagenome and their relationship with the report, relationship to others, and so on. But in addition, then this goes into a major population data bank. But in late March, actually very quickly changed their sampling protocol. They went from dry swabs to 95% ethanol, and then they could preserve fecal viral RNA. It's not that good for microbiota, but it's still okay. It's not as good as some other things, but still okay. But then with the possibility of having the virus preserved was really important for them. And then they actually introduced a small finger stick blood sample in the same kit. And I assume they will use it to detect antibodies. So suddenly they have the opportunity with this kit now to study active and previous infections or COVID and microbiota. So, but maybe it's not the microbiome that there are also other things which can and should do. And I thought we were actually going to end this talk by Marius going through the kind of one of the largest things ongoing at the hospital related to COVID these days, which is the WHO Solidarity Trial.
Yeah, I think just to follow up what you said about Segal and Rob Knight, I think without comparison, I think also our research group has also tried to adapt to this situation and try to focus on what is most important. So Hanne, for example, she has got time off to do biobanking at Ullevål, has done a tremendous job there. Hedda Hoel, she's also a PhD student. She has taken a break from her research to go to Lovisenberg and work clinically, and also to collect people to this trial. So this, and personally, I'm part of the national coordination team in this WHO trial. So next slide. So this, you have probably read about this trial in the newspapers, but I think it's a quite unique trial because it's a randomized adaptive trial. So the idea is really that we can try different drugs that have been used in humans for other indications. And all these drugs have also been tried for a SARS-CoV-2 virus in cell cultures, and they have been effective. And the endpoint in this WHO Solidarity trial is really hard endpoints to see if any of these drugs can improve survival. So next slide. So if you remember this slide with the virus entering the cell, you can try to inhibit this on different parts. You can inhibit the protease when it enters the cell with this drug called Camostat that's being tested in Denmark in a randomized trial there. You can try to work with the endocytosis process, and there have been some reports that these malaria drugs, hydroxychloroquine, can alter the pH of these endosomes and thereby perhaps inhibiting the virus. And then you can be more direct, and you can target the RNA-dependent polymerase, and that is what is being done by remdesivir, which was tested previously for Ebola virus. Next slide. So in Norway, we have rolled this study out in 29 hospitals, and compared to the WHO core protocol, we have a much more detailed setup with biobanking. We have 3-month follow-up with questionnaires, with lung function tests, with echocardiography. We'll also take fecal samples there. And at the right, you see the steering committee. So it's a very nice committee. I think we have a statistician in the committee. We have a pharmacist, which has been very important. Next slide. And the inclusion was very nice and rapid in the beginning. You see most patients have been included from Ullevål, Bærum, and also the other Oslo hospitals. And then a few patients have been included from the other parts of the country, but this really reflects how the pandemic has hit Norway differently. Next slide. So we also tried to get Sweden and Finland into a common Nordic platform where we test the same study drugs, we use the same ECRF system, and we have the same system for biobanking. Unfortunately, this process has gone a bit slower in Sweden, especially because of the regulatory system there. So they haven't really started yet. Next slide, please. But in Norway, we started end of March as the first country in the world, together with Spain, started with these malaria drugs, hydroxychloroquine versus standard of care. And then a week later, we started also with remdesivir. It was a bit difficult to get it into the country because it's a completely new drug. But we have randomized also between 15 and 20 patients now to remdesivir. Next slide. So the first, until beginning of May, we included more and more patients every day, but this curve has really flattened out now. So now maybe we recruit 2 patients per week, something like that. Next slide. So the challenge we face now, not only in Norway, but also in the rest of Europe, is declining inclusion rates. This WHO study is also going on in Brazil, so there we'll probably have better inclusion. There's also a lot of noise going on. There's a lot of data presented often as press release before peer review and publications. And then we have to discuss this with the investigators and patients and so on, because they read about it in newspapers. And then last Friday, there were actually some solid data both on hydroxychloroquine and remdesivir, which might have implications for the study. This is a registered trial that was published in The Lancet. I think it's the first time I've heard of a registered trial that temporarily stops an intervention trial, but it's very solid work. It's 96,000 patients, 15,000 of them have taken chloroquine or hydroxychloroquine alone or with macrolide antibiotics such as azithromycin. We know from before that both these malaria drugs and these antibiotics, they can alter the ECG, the QT time, and they can cause arrhythmias. So in this study, in the standard of care arm, there was 0.3% who had these ventricular arrhythmias. In the hydroxychloroquine alone group, there was around 6%, and when you combine with the macrolide, it was 8% who got these very dangerous arrhythmias. And there was no benefit of taking hydroxychloroquine or chloroquine. And of course, this is a registered study. It's not randomized, but still the signal is so strong. So you can go to the next slide. That the WHO Central Study Group decided to suspend the hydroxychloroquine arm until data have been analyzed. So data from not only our study, but also the Discovery trial in France and other trials will probably pool the data now to conclude the hydroxychloroquine arm. And if there is no effect, it will be stopped in this study. The remdesivir arm will continue. There was a study on last Friday from this NIH study, which was finally published in New England Journal of Medicine. It's very strong data, but it probably works a bit. It probably reduces mortality with around 4%, but it's not statistically significant. So we need to continue until we have real data. So the trend now is to pool data from different studies, to coordinate studies, and to plan for the next phase. So the solidarity is maybe now going to be fused with the French discovery trial, and we are seeking for EU funding to achieve this. And from Norway, we have been working with a future adaptive master protocol, where we try to be able to test new drugs in phase 2. Here we will have skewed randomization, so we can put almost all the patients on new drugs, and then we have historical controls in the phase 3 arm. And then in the phase 3 arms, we can really see if these drugs are working on hard clinical endpoints. Yeah, that's it.
Thank you, Marius. That's a nice overview of the Solidarity trial. There was just one thing I had written wrong here. So the remdesivir arm is definitely still ongoing, right? Because it's promising. But you'll do a preliminary analysis so far just to see the status, or is that just the hydroxychloroquine that will be analyzed now?
The hydroxychloroquine arm is going to be analyzed this week and then concluded. And the remdesivir arm is probably going to be analyzed mid of June. Then we probably have enough data to conclude there.
Right. So this actually brings us to the end. I'll try to summarize what you said here. You might have learned less on the microbiome that you thought, but I guess the idea was here to show that this virus, SARS-CoV-2, is definitely infects the GI tract and it replicates there. It causes GI symptoms and is shedding in stool, whatever that means for us or for anyone, we don't know. But so far, there's really no data on the impact of the microbiome in this setting or whether the virus impacts on the microbiome. I think this, as you have all experienced, it has had major influence on life, but definitely also on research activities in our field, but also other fields. You know, we have to help cure this disease. So it definitely is an opportunity to reorient and it's critical to contribute where it is important. I think try to view, when you do this, I think it's important to look at this as a way of like science pays back to society, right? So we have to try to help out now. And it really seems like science does it. Although there's really also lots and lots of noise of not that good science and so on. So that's a challenge in that regard. But remember then that our usual patients and research definitely still need our attention because there are many, many of them and they are actually very afraid of what's going on and how the virus will affect them. And that's pretty much it, I think. Thank you.
Yeah, thank you.
So I haven't got any more questions, but now it's actually open to ask questions or have comments. Okay,
so from Siv could the lack or effect of hydroxychloroquine depend on the timing of when you start? Yeah, of course, that could be a point. I think if you look at the influenza studies with Tamiflu, you need to give Tamiflu early before the virus is spread from the nasopharynx to the rest of the body. So that could definitely be one point about the hydroxychloroquine.
What's the role of these anti-inflammatory drugs, anti-IL-6 and anti-IL-1 and things like this?
Well, there have been some reports on anti-IL-6, again as press releases. There was a French group claiming that it had significant effect, clinical effect, but they did not give any specified endpoints. And I've discussed with French researchers now for our collaboration, and they are not so happy with this way of presenting results there. So I think it's not conclusive. I think if you want to do immune modulatory treatment, I think targeting IL-1 system is with anakinra is probably the safest. It's a short half-life, and there was a nice overview in the Lancet Rheumatology a few days ago, going through the studies that includes that. So, for example, the biggest study, ReMapCap from Britain, has an arm with anakinra. So I think that would be very interesting.
We got the question from Trine: are there other unknown receptors on ACE2? And I'm not. I think if they are, there are unknowns. So I don't sure we know. I think, but the viral entry replication cycle, I think that's very interesting actually, and the role of these cleaving the cleaving enzymes, the TM, pre-RSS, the cathepsin, things like this. That's of course where there is of interest.
I think this I think this drug Camostat, it's a very old Japanese drug that was it has been around since the 80s. I think it has been used for esophagitis, for pancreatitis, and so on. It's a serine protease inhibitor, so that interferes with the cleaving step, and I think that's interesting. There have been also some old results showing that it also inhibits inflammation through NF-kappa B signaling. It could also inhibit this system that we heard about in the early 90s, this kallikrein-bradykinin system, which is also very interesting, because if the ACE2 receptor is blocked by the virus and not longer available, then it could be too much bradykinin, which could cause pulmonary edema. So this Camostat, I think, is maybe the most interesting of the not yet studied drugs.
Okay.
There are some other questions.
Do you look at them?
Regarding anakinra and tocilizumab, there are no solid data on microbiota. I'm not sure. Do you know that?
Well, I don't know. Anakinra, goes into the inflammasome story where there's lots of data on microbiome, but I'm not sure whether actually the drug, to what degree the drug had affects it. It could be. I think it's a good. I'm not sure.
It's a good question. It was used in the CANTOS trial where it reduced reoccurrence of myocardial infarction, but I don't think they collected any stools. I'm not sure.
And then Brian asks again, if we look at the cleavage enzymes, does this also correlate with ACE2 expression? And I don't know actually whether these whether these are ubiquitous, whether they are circulating, cell-bound, etc. But so whether they also could contribute to the tropism of the virus.
Yeah.
In principle, they could, because if you have cleavage enzymes in the same cell as you have in the cells where the virus replicates, then there's a theory then that the virus could then be activated on as it's being released. So that would really facilitate viral replication. But I'm not sure what's known and what's hypothesis and so on.
But the ACE2 seems to be, at least I haven't read all these reports in detail, but it seems to be related to dysbiosis in some studies. I haven't had time to read all these papers in detail, really. So, but there's something worth looking into there. I also try to see if it's possible to look for microbial pathways that could be linked to ACE2, but I'm not sure, Johannes, in pie crust and so on, if it's possible to predict ACE2 expression and so on. I haven't had time to look it up.
No, I don't know. But what is I think one interesting question or kind of way of looking at this is we observe a very strong age trend in severity and mortality, and you have a male gender. So these are, at this age, is known to influence microbiome. So that could potentially be a hypothesis that microbiome changes are related to age, increased susceptibility to serious infection. while male gender is so far, I'm not sure how well explained it is. Many theories, one of which of course is ACE2 expression, since this is actually encoded on the X chromosome.
It was interesting, this figure you showed, Johannes, with female and male ACE2 distribution, where in women you see it's also distributed in, it seems to be in the subcutaneous adipose tissue. Could that act like a shield or something? It's just speculation.
I don't know.
No, it was just, it's the first time I saw this figure yesterday night, so I was just wondering if this could explain some of the gender difference, but it's just a question.
Yeah. So there are many opportunities there. Okay. We haven't got any more questions. I guess it's time to start closing the meeting.
I think it was very nice that so many could come and participate in this event. I mean, it's I think it's, even though it's COVID everywhere, I think it's nice to also keep this network going and also to interact. Yeah, so thanks for that.
We also mentioned that we are planning national conference in November. So we'll just have to see. It has to be according to guidelines. So we just have to see how many we can fit into the venue, whether we should change the venue, whether it has to be digital, or we don't know. But we really want a conference with scientific contributions from you guys. So we will announce this before summer, at least the abstracts and so on.
Yeah, and we also have a network meeting in June, right?
June
16th of June.